Okay, now the previous one was number 4. I'm jumping to 6 rather than 5. They are practically on the same level, but it makes sense logically to deal with this issue before the number 5. Reason 6 is for allowing plants to adapt to climate change, and number 5 was allowing plants to adapt to other environmental change, such as fragmentation and environmental weeds and a whole range of other things. Now, I'm going to return to my little wallaby grasses, which are over on the table there. I'm sorry I wasn't able to show you with the camera what they look like, but they're over there if anybody wants to look at them. They're doing just fine. They grow very much more vigorously in lowlands, in pots. They flower at any time of the year, whereas in their natural habitat at the top of the highest peaks in the Victorian Alps and into Mount Kosciuszko, I don't think it's in Tasmania, it only has a very brief growing period each year and doesn't produce as many seeds and so on. So it really likes being down here. Now you're thinking, well, no wonder, I'm treating them nicely, I water them, I put them in pots and all that sort of stuff. But the point here is that a species of organism, whether it's a plant or an animal or a fungus or a microbe or whatever, occurs in a particular space, not necessarily because that's where the conditions suit it best. They occur where they are able to fit into an ecological niche with all the other flora and fauna, microbes, fungi, and the whole works. And so a large proportion of species, and I don't know what because I'm not a geneticist or whatever, a large proportion of species occur in locations which are not their most ideal ones, but where they can occur. Now, if we're thinking about things like mountain pygmy possums or the crag wallaby grass over there, they're on our highest mountain peaks in Australia, and you would think that they are the species that are most prone to disappearing in the face of climate change. Nevertheless, the crag wallaby grass and all my other alpine wallaby grasses, of which I've got probably 15 species growing, they all do great guns down in the lowlands. You give them warmer climate and they love it. So it isn't that they particularly like the cold conditions, in the case of the crag wallaby grass, jammed in cracks between the rocks. It is that that's where they can exist without getting outcompeted by other species. So when we're talking about adapting for climate change, I think we need to bear in mind that the species of flora and fauna that occur in a particular location may be not limited to their current situation simply because they need that soil type, that level of soil moisture, that climate, all those sorts of factors. It may be simply that they fit in with the other species that are there. And between ice ages and interglacial periods, these things are able to hang on, even in situ. And I'd like some of the geneticists perhaps to address that issue about things like, I raised the issue of Garnia radula before in the Grampians. I reckon it's probably been the same genotype in the Grampians since it would have been, well, since the last ice age, when it would have been covered with snow and ice for a lot of the year and so on. But that species is able to persist there. And it would have had a whole lot of other species which would have persisted there. And I think something like the crag wallaby grass, and I'm very keen to hear what the geneticists say about this, I think on an ecological basis, as long as there aren't new species that suddenly rush in, and we're told that climate change is so fast that species aren't going to be able to to move in very rapidly, there's still going to be the same mix of species. There are a whole lot of species that grow with the crag wallaby grass that also do perfectly well in lowlands with a warmer climate. So I'm left thinking, well, I'm not as concerned as I might have otherwise been about something like the crag wallaby grass disappearing in the face of climate change, because I know it can do perfectly well with warmer conditions. And as an ecologist, I think about how it fits in with the environment, and the thing that worries me is whether the other species that it's occurring with are able to adapt, and how many of those are going to persist and how many of those are going to disappear. If other ones disappear, maybe the crag wallaby grass will take over and do even better, despite it being confined currently to the snowy regions. So I bring this up as an issue, and I'm really keen to find what the geneticists say about it. So can somebody on the panel address that sort of issue for me, please? Ari again.
So this is So people have done a lot of modelling around Australian plants in recent years and asked, well, what's going to happen under climate change? Which ones are going to benefit and which ones are going to actually not benefit? And clearly we have winners and losers, but we have an awful lot of losers. So this is work that Craig Nitschke has been doing on Eucalyptus regnans, and here we have a situation where mountain ash on the left-hand side, that's its historical suitability, and on the right-hand side, of course, that's the predicted distribution under climate change. So we have these sorts of examples, and this is actually based on physiological data that's then put into a model. And of course, one of the challenges we have is that under climate change, it's often the extremes that matter. So in other words, you might be okay in a lowland habitat, but of course then you get a drought that comes along, and then it of course can take the plant out. And I'm sure people here have done plantings of that nature. I've certainly had brachyschome growing from the alpine area, and then of course they're great as long as they get watered. As soon as it gets too dry, that's it, they're gone. And of course lots of other daisies are perfectly okay under dry conditions. So it's the extremes that really are important, and there are certainly lots and lots of modelling exercises out there that clearly indicates that we are in trouble under climate change. We do expect dramatic changes, notwithstanding the sort of comments that Graham is obviously mentioning, into the future. It's not going to be a stable environment. And of course in Victoria we have some examples of that already happening. So again, this is in the alpine ecosystem, and this is work that Henrik Wyron did across the years. This is one of the few long-term sites we have in Victoria where we've been monitoring various species across many years. And this goes back to plots that were originally set up in the 40s. And clearly we've got a situation where we're seeing quite dramatic changes in the grasses versus the forbs versus the shrubs. Some are going up, some are going down, and those changes are happening in a few decades. So we are in a period of rapid change, and that's the first thing that I think we really do need to bear in mind when we think about these sorts of issues.
So I just wanted to say the same thing. There is rapid change, there's winners and losers. And we recently did a big genetic study of Meliodora, Eucalyptus Meliodora, from Queensland right down into Victoria. We can't find any evidence of genetic structure, which was intriguing in itself. But then the more important thing we did was to model future climates for that species and then overlay available habitat based on today's land use. And a couple of things that came out that are serious for this species is that there's no land for it to move to. So even if we could pick a provenance and move it around, the actual places for planting are diminishing because most of the places we need to move it to are already taken up by urban or agriculture.
Don't go yet. Now, okay, we'll get rid of that. Okay, they're good points to make, but I've got still this concern that when you talk about finding where they're going to be able to occur, like the mountain ash or the yellow box in future time, it's done on the basis of modelling where you look at climate and soil type and all that sort of stuff. It's not looking at this aspect that I'm talking about, that a species occurs at somewhere, not just because the soil is right, not just because the moisture is right, it is there as part of an interconnected ecosystem. And if we are to work out where a species can occur in future, we need to anticipate the other species that would be growing with it. So with climate change, for example, it isn't just the soil type and so on, it's things like fire frequency and stuff like that. And that in turn depends on other species that are there. And we don't know what all the other species are going to do. How many of those are going to disappear, how many of them are going to benefit, and hence how the whole system is going to change, and hence whether perhaps a mountain ash or a yellow box may be able to occur in conditions outside the modelled distribution, which is based on the current relationships between the species at all those different locations. So to what extent do the geneticists think we may get a different result if we were to predict the future zones for a species, taking into account all of the ecological relationships? Were we able to do that?
Sorry? Small question, keep it short. I'll try and keep it short. I guess my view is that in 50, 100, 200 years, what we currently think of communities will not exist anymore. There's going to be a massive change in the way things restructure themselves. And I think genetics can help us predict perhaps some of our most vulnerable species, how they can respond. But I think there will be wholesale rearrangement of what we currently see and what we currently love as our communities, and we're going to have to actually come to terms with some of that change.
In some ways, Graham, you were asking an ecological question rather than a genetic question. So, no, let me just say two things. So, firstly, the original models that people were doing was based on correlative approaches, where you basically say this is the current distribution of a species, what climate factors does it correlate to? Then you say, well, how much is that climate going to change? And let's then predict the future distribution of that species. That's a correlative approach. These days, ecologists are tending to move towards mechanistic models where you actually understand the organism itself. So, for instance, in that regnans example that Craig Nitschke's been studying, what he's done is he said, well, let's look at the physiological tolerances of the regnans and say, well, what can it tolerate, what can't it tolerate? And then we look at the future climate and then we decide whether it's going to fall over or not. So we really are developing much better models of what we think is going to happen in the future, and we're not there yet. I'm not disputing that. What people are also doing in their models is to consider the biotic interactions more and more. So again, initially the models were based on considering a single species, but now, of course, we're starting to look at whole communities and the interaction among those communities. Now, we still have a long way to go, but it doesn't change the fact that these communities are going to dramatically change. So, for instance, in the Alpine area, people like James Kamak have been doing models where they've basically said, well, what's the consequence of having shrubs basically moving in? And, of course, what happens is that you then predict a more intense fire because shrubs burn much more intensely than grasses. And what you effectively get is a feedback loop where, of course, you get the more intense fires, and the consequence of those, the shrubs will actually also establish more easily. So as a consequence, you're getting shrubbier and shrubbier as a consequence of the interaction between the fire and the climate change. So I think we have a long way to go. I think we're getting better at making those predictions. We're certainly aware of the limitations of the earlier studies, but we can build up knowledge in this sense. And it doesn't change the fact that communities are predicted to change under these different models regardless.
I'm Sharon Mason, I'll do. When you said Eucalyptus mellodora, no evidence of genetic structure, what do you mean by that?
Oh, okay. So genetic structure is one of the ways we try and figure out historically whether there are zones that are more genetically related than others, and that might tell you not necessarily that there's local adaptation, but it's certainly an interbreeding population. So that might give you some idea about collection zones. So if you've got structure, if you've got populations that are grouping together according to some geographic region, you might then go, well, we're only going to use seed from this region, and someone else might go, we're only going to use seed from that region. So that's what genetic structure is. It's like a historical view of genetic variation over time. I can see you're still puzzled, so I need to keep going. All right, so if you imagine, I've surveyed a suite of populations for yellowbox. What I might have expected was that some populations have been isolated from other populations, say by a mountain range. And they've developed their own different gene pools. Yep. What we didn't find any evidence of that with yellowbox at all. And we think that's what that's telling us. We think that that was once one very large interbreeding population where there was plenty of dispersal across the population historically, because these were very old trees we surveyed deliberately to look at that historic structure. Does that make sense now?
Right. We have some Vox vote questions.
So one of the questions that's come from the audience is that there's a lot of work being done on biolinks at the moment, and could the process of creating habitat connectivity and biolinks alone be enough rather than kind of facilitating something else? Could the process of relinking habitats be a good way of kind of dealing with these issues?
Connectivity is important, but let's not forget that some of those processes are fairly slow, so help still, from us can still be required. But I want to kind of step back and get back to the whole genetic issue. I think there is, in general, a little bit of a misunderstanding or misinterpretation of the type of information that we can obtain from this genetic data. So genetics in this context really provide an ecological tool on how to understand demography, dynamics, and patterns across the landscape. So they help us understand the connectivity across landscape and across geographic features, such as what Linda was just mentioning ago. They can understand the connection, the association between population size and diversity and fitness. They can help us understand how to interpret current patterns across space in relation to events that happened in the past as opposed to events that are happening now. So they give us a better understanding of ecology and evolution of the species, of dynamics and demographics. Adaptation is a very different question. We can make some guesses and inferences, or we can do dedicated study to explore that particular question. But it's much more difficult to get a definite answer on that. And also the other thing to keep in mind is that this is one of the reasons why mixing genetic diversity sometimes is good. As Graham's saying, the more genetic diversity we have, the more we have the capacity to include within the system this type of plasticity that is, let's not forget, not always controlled by the genes, but it's often controlled by the environment as well. So it's the connectivity between those two elements that needs to be kept into account. Did you want to add anything to that?
So BioLegs came out of some thinking that was done more than 10 years ago, and certainly DELP, or whatever they were called in those days, I forget, supported it quite strongly. It was in the 2009 Biodiversity at a Time of Climate Change white paper, which came out of the green paper. And what people then did was to draw a series of links across the maps, and they were saying that's going to be an excellent way of actually adapting in situ. And I think in terms of promoting gene flow, particularly in animals, that's going to be a pretty useful thing to do. The problem is that, of course, when you think about Melbourne's climate looking like Wagga Wagga in about 30, well, could be 30 years, hopefully it'll be 60 to 80 years, but you simply don't have enough time to get gene flow happening to bring those pre-adapted genotypes down to Melbourne, given that that's the climate that we're dealing with. It's a great idea, it'll work well for many animals, but we have a challenge if we have species that have a long generation time.
So I'll just go through this quickly because we've talked about this a lot already, but I think there's two big concerns for restoration projects. One is this idea of provenance, and one is capturing genetic diversity, and we've talked about that in terms of inbreeding and outbreeding. So just to put your mind at rest, provenance is not a genetic or an ecological term. It's actually from the art. So it came about through French, through the French art trade, basically. It's come to mean a lot of things. It's still important as a term in forestry. We talk about it a lot these days. But what it has driven is this idea that local plants do better than non-local plants. You know, they're more adapted to the local environment. We've talked about, what actually is local, and there's genetic theory as to why local plants would do better. But is that really true? And that's why we're here today. If so, if it was really true that every plant was really locally adapted, we couldn't move them around anywhere. They couldn't go very far at all. So it's clear that, and we've had having this conversation, they have some plasticity, they have some capacity to move. And selection for genotypes, which is where we're getting to now, is not just about climate. And as Ari's talked about, when we're modelling early models, we're using climate as a surrogate for lots of other variables that things might be adapted to. And we've talked a bit about soils, altitude, but think about pathogens and herbivores. That can also drive strong selection for local adaptation. So something might be locally adapted because it's resistant to a pest, not because it's so in tune with the environment. And then Ari's talked about this study that was done some time ago, the meta-analysis that looked at local versus foreign, and they looked at it in two sorts of ways. So they gathered all the information up they could in 2008. That was 32 species. Now that's not a big pool of studies to choose from. The reason it's so small is because it was 32 that they could compare. So there's lots of data out there, but doing a meta-analysis means that you must be able to compare the studies. So they got down to 32, and they looked at those that looked at local versus foreign at one site. But to truly understand local adaptation, you need to do the reciprocal transplant as well, which is the one on the right, where you actually do the two, local and foreign, in two environments. And Ari's already told you this, 71% of the transplant sites, the local did better. But interestingly, that local adaptation was more common in large populations, not small populations. And that's because small populations are often showing these issues of inbreeding. They have limited ability to adapt. They just don't have the genes in the gene pool. When a strong selection event comes along, they often get wiped out or down to one or two. Then they go through a genetic bottleneck and have got to recover from that. There's no time for them to adapt. The other thing out of this part of the analysis was that they couldn't make any predictions. So local adaptation didn't seem to be related to how long the species lived, what mating system they had, and whether they had some sort of spatial or temporal variability. They also found that geographic distance, and they were looking at studies from 3 metres to 3,500 kilometres, didn't influence the strength of that local adaptation. So if you were 3,500 kilometres apart and brought together, that didn't mean that your result out of the experiment was any stronger than if you were 10 kilometres apart. And what they did find, though, and I think this is really significant for this sorts of discussion, is that geographic distance and environmental difference were the key indicators for whether they were going to find locally adapted populations. So that's interesting, 71%. But if you do the reciprocal transplant experiment, that actually drops back down to about half. And that's because some plants did better in both locations. It didn't matter where they were. So this is why you might find we're a bit hesitant about giving you guidelines and telling you how things are going to perform, because it's quite unpredictable in the space that we work in as researchers at this point in time. I think the other key thing that came out of this is that the evidence for plants, or foreign plants, being maladapted to the environment they got put into was very low. So that doesn't seem to be a real issue. We often talk about, don't bring things in because you might be wasting your resources. This study is showing that that's very low. So their conclusions was that local adaptation is less widespread than previously thought, which I think you can all now breathe a sigh of relief. It's not as much as you think it is. There's a range of studies now coming out. Ari's mentioned a few, but there's a whole range of other studies coming out about Australian species, because they were all foreign species in that previous meta-analysis. I can give you these papers if you're interested in reading them. I can give you the links to them. A lot of it's actually showing little evidence for local adaptation. When it does, it's kind of like for the button wrinkle wart, it's kind of in interesting traits, like the local survived better, but the foreign grew bigger and had more flowers. What does that mean? If you want a plant with bigger biomass and more flowers, put foreign ones in. If you want ones that survive better, in this case, it's local. So that's quite interesting. I think one of the most interesting things is some of Nola Hancock's work and some of Nick Gelly's work is showing that actually the local provenance have the highest mortality for a couple of eucalypts they've looked at, and actually have the highest herbivore and pathogen stress. So, and that's what actually keeps them in check in the local environment. If you think about this holistically as a system, the reason things don't go rampant and weedy is because things are keeping them in check. In terms of how you might choose your provenance, there's lots of suggestions out there, most of which are untested at this stage. Very complex ways of looking at how to choose your provenances. I've starred the 3 red ones because I think they're your best bet hedging. So they give you some sort of hedge against only going local under climate change, going completely non-local under climate change. The admixture, composite and climate adjusted actually give you a bit of variability that you can put in and the plants let them sort themselves out over time. To choose which of those, Martin Breed has actually come up with a decision tree. It doesn't have, can I make that? It doesn't have the climate adjusted, which is the most recent one, on it, but there are some ways to work yourself through making decisions about that. It's incredibly complex if you're dealing with multiple species because they might all need a different strategy, so that's going to add time and dollars. But it's worth having the thought before you go out collecting seed whether you want to follow one of these. And Suzanne Prober and I are actually trialling the climate adjusted provenancing with some NRMs at the moment to get some plots in the ground to see how that actual strategy works out.
Yeah. You just said there you were talking about a eucalyptus species, and you said the local ones kept in check by pathogens and herbivores. And isn't that one of the points, is that you can bring stuff in from outside that seems to do well, but the reason they're being held in, the local ones have been held in check, is because they're for local species of herbivore and pathogen, which are still important parts of our environment.
Yep.
And that's the very reason, maybe, why we should be planting them. Because if you bring the ones in from the outside and they're not being held in check by those herbivores and pathogens, where do they live?
Yep.
And that's a sound argument. So I'm going to give you a different example. Is that eucalyptus? So the question was, should we not stick with the local? Because I said that they were more being kept in check by possibly herbivores and pests and diseases. So is that not a reason to keep the local, whereas the outside ones are not being kept in check? Have I got that right? They're not providing habit. Yeah, that's right. Yeah, that's right. So they're not providing the functional resources, so the resources that those pathogens, herbivores, diseases might need to keep them going. Yep. So Eucalyptus viminalis on the Monero, just north of Cooma, is completely out of the system. It's almost dead, and we have no opportunity to bring local in at all. So in that case, we're actually setting up trials to, we've managed to salvage some local seed off some isolated trees, low genetic diversity, but we're actually going to go as far away as South Australia to a different subspecies to put in some trials to see if this is a species that can no longer exist there, what's going to be the replacement species? What can we think of might be a suitable provenance to replace what's there? And because we've gone over a very, very broad range to collect, we're hoping that at least something is going to survive into the future. We're also putting in a completely different species, just to throw that one out there. We're actually putting in quite a rare species, Eucalyptus benthamii, because it's incredibly frost tolerant, and we think that it's got the attributes required to survive in that now very open area, and that it will be able to cope also with the change in climate as well. Don't know, but we're going to put it in and give it a go.
Yeah, no, that's all right. I'll introduce myself as a ring-in. So my name's Rebecca Jordan. I've just finished my PhD with Ari Hoffman, as well as Suzanne Prober and Shannon Dillon at CSIRO, looking at genetic diversity and adaptation and think about how we can use them for restoration. And so what Ari's sort of thrown to me is talking about this idea of climate future plots. So as it seems to be coming up again and again today, is this we have some data, we have some ideas, we have some things that we think may or may not work, but it's really that empirical data of exactly will this work, how will it work, that we really lack at the moment. And so one of the ways that has sort of been proposed to try and address this is something called climate future plots. So we're thinking about, within our restoration or other projects, actually setting up experimental plots to test this idea of provenancing, to test, can we move things from different provenances? If we move things from warmer areas in, will they survive better? Will they survive better when we have a heat wave in 10 years' time? To try and actually get some data behind this. So similar to Linda had some really nice graphs up of some proposed provenancing strategies, and the ones that she had hedged bets were this idea of not dismissing local, certainly considering local, but then also considering sort of wider collecting as well. And so that's really what, if we can bring them up. So that's what the climate future plots are trying to think about. Yeah, there we are. Perfect, beautiful. So when we're thinking about the climate future plots, what we're trying to think about doing is going, okay, well, we want to consider putting local in. So the local, certainly in these graphs, is the black star. And so the blue dots are thinking about where we might collect from in the landscape and how much we might collect from at those areas. So what we're doing then is actually setting up an experimental plot. So we're putting in our local, but then we're also, say, collecting from, I'm not sure, say, 50 k's away, 100 k's away. Say if we're thinking about certainly the species I worked on, which is greybox, Eucalyptus microcarpa. Maybe we're planting in Bendigo, so we take some from Bendigo, we take some from wider Victoria. Then we think, well, maybe it's going to be warmer, so let's try getting some from Wagga and some from Dubbo, which are sort of projections for sort of 30 and 50 years' time, and putting them in. But putting them in such a way that we know where they came from and where they went into the site. And then this is what was said earlier, where that recording becomes really important, that it means in 5 years' time we can come back and see which provenance is doing better, which is doing worse, which is maybe doing too vigorously. And then we can come back in 20 years' time after a heat wave, who's still performing well now? And actually, in doing that, hedge bet, get some of that diversity in. So as a population, as a whole, we have enough diversity that something will continue surviving, but that we can also get empirical data to help keep informing the restoration as we go along. So I think, have I missed anything there, Ari, or is that...
One point that's worth making is that we're not saying you should take everything. You keep most of the stuff local. So most of what's going in should be local, but basically you allocate a small proportion of your land that you're going to revegetate for one of these plots. That's what we're saying.
Yep.
So it's in a sense it's an insurance policy, right?
Hello, Lynn Meredith. I work as a land management officer for Manningham City Council, and we've done lots of revegetation projects, and everything's been eaten by deer, so we're going to deal with them. But when that's dealt with, and that idea of moving things in the landscape, is it not plausible that we should just be taking red box and be planting it down in the gullies in the swampy areas and seeing what happens? And I mean, is that just too wrong?
That's a super good question, and I think that's similar to what Linda was just touching on, this idea of moving. So, here we're focusing on moving different provenances around, so we're staying within a single species, but then certainly the natural question that follows from that is, if we're talking about areas that are going to become totally inappropriate for that species, do we need to think about filling that ecological niche with something similar but a different species? And that's a really great question that, I don't have an answer for, but is a very great question to be thinking about.
Thanks very much. Irene Kelly from the Knox Environment Society. So the plot that you're talking about putting these different type trees in, is that like the ecosystem, a defined piece of ecosystem that would normally support those trees, or is it a revegetation site where you're manufacturing the associations of plants? Or are they just going to be growing on their own?
Yeah, look, I think it could occur on any of those scales. I suppose the way I was sort of describing it is thinking about it in restoration projects, so where you're focused on maybe one species or maybe a small subset of species possibly, then a whole ecosystem, and that's where I envisage them occurring more. I don't know if Ari can comment on that more, but I think it's more in that revegetation space than necessarily a sort of a native ecological environment. Yeah.
Okay.
It's kind of
Yeah, sorry.
It's kind of interesting because that's how we envisaged it in the first place, and that's how other people have talked about it. And I should just point out that these sorts of plots are being set up overseas. So it's not like we're doing anything unusual here in that sense. But what we're also getting feedback on is people that work in intact systems, and they say, well, this could be really useful if things start falling over. So all of a sudden we do see a lot of these trees dying. Maybe that'll give us some options in terms of then the climate future plots acting as nursery sites for the future, even in intact ecosystems. I mean, we hope we don't have to do that, but you never know.
Thanks very much, Rebecca and Ari. Any other questions on those sorts of things? We will be looking at, yes, up the back right, please, Warren. Oh, there's two.
So in terms of getting the results, there seems to be an issue of time. And is it possible that in the amount of time that it takes to get these results, will they not so much become irrelevant, but will we have the time to implement them before we need to look at even greater changes in the environment? So after 20 years of looking at a plot, are there going to be greater changes than what we're seeing? And can we use that data that we get?
So Linda, you should probably take that as well. But I think what we want to do is we want to obviously monitor because we want to learn as we're going along. And you're saying, well, it could be too slow, and that may well be the case. But what we're trying to do here is to, of course, then say, well, we're not taking anything... Sorry, we're trying to take some stuff fairly close by, some stuff further away. So hopefully what we're doing there is minimizing that particular risk. But I agree, I mean, the rate of change is going to be very fast. That's what we're facing. But, that's the best we can do, and at least it's a lot better than doing nothing. That's how we feel about it.
Yeah, that's absolutely right, Ari. We can always say with hindsight we should have done something different, but this is an opportunity, I think, to get on the front foot and try some things before it does get too challenging to do this. The other thing, of course, some of these could end up as being seed of the future, depending on how the genes mix and what sort of types come out of it. Potentially, that's what we need to be, planting in 50 years' time, will be these complex mixes that we've actually put in as these plots that are being monitored, and we know what they're doing.
Wonderful, thank you.
Another question
up the back.
Hi, Kim Saunders from Yarra Ranges Council. We've been contacted just recently by the Hawkesbury University, and they're looking for some trial sites, 2.5 hectare trial sites to test some of this stuff. I'm just wondering if you guys or anyone in the room has been involved in that, and whether that conversation is going broader than here.
Yeah, Ari and I both know about it. Rebecca probably does as well, Maurizio. Yeah, we do know about that's Paul Reimer's group from Hawkesbury. So a couple of weeks ago, Ari, Paul and a few others, we all came together and actually started talking about the plots that we're all starting to put in, and can we actually develop them into a bit more of a national framework for doing these sorts of experiments. So we are all talking to each other. All of our experiments might be slightly different, but we're going to try and look at actually layering at least one level of similar experiment across all the work that we do. So we are all in contact with each other at this point. Yep.